patulin level in apple juice. A part of apples destined to juice production is generally of poor quality. Penicillium expansum is the most important fungal species causing spoilage in cold-stored apples. P. expansum produces patulin, a toxic secondary metabolite. Growth of P. expansum on apples via wounds results in necrosis of apple tissue. This may result in contamination of the apples or its products, for example apple juice, with the mycotoxin patulin. Patulin is harmful if injected. It is mutagenic and causes neurotoxic, immunotoxic and gastrointestinal effects in animals. Moreover, patulin is a potential genotoxic and can induce oxidative DNA damage in human cells causing mutagenesis and cancer. Spores of Penicillium are soil inhabitants. They can thus contaminate apples pre-harvest from soil. Handling of apples can then result in wounding of the apple skin. Contact of such wounds with spores of P. expansum can lead to rapid germination in the tissue. This increase in inoculum size may influence in rot development and patulin accumulation. The US FDA has established an action level for patulin in apple juice of 50 micrograms/kilogram or parts per billion as determined on single-strength apple juice or reconstituted single-strength apple juice. The study using storage treatments. Number 1. 2 isolates of P. expansum, A-delta 3.72 and A-delta 3.78, obtained from cold-stored apples. They both were proven patulin producers. The isolates are stored in plugs of potato dextrose agar, PDA, submerged in distilled water at 41 Celsius. Number 2. In total, 84 locally produced apples were used which had previously been cold stored. Number 3. P. expansum was grown on PDA at 25 Celsius until sporulation occurred. Number 4. Apples were surface disinfected in a solution of 2% sodium hypochlorite for 1 minute and rinsed with distilled water. They were then wounded. Number 5. For both assays, half the apples were placed in a storage room at 1 Celsius plus or minus 0.5 and 85 to 90% of humidity. The rest were incubated at 20 Celsius. The lesion size of the apples incubated at 20 Celsius was monitored and recorded daily or as required when lesion size was about 2.5 centimeters. Apples stored at 1 Celsius were periodically checked until fungal growth was observed. When this occurred, lesion size was monitored daily and recorded. As before, rotten samples were obtained when lesion size was about 2.5 centimeters and patulin contamination levels were analyzed. Number 6. Each apple was weighed and the whole necrotic tissue area was removed. Patulin that may have diffused to the sound tissue was also collected and total patulin in the apple was recovered. The samples were weighed and frozen at 25 Celsius until patulin extraction and analysis. Number 7. Distilled water was added to the samples in a 1-to-1 ration of weight relationship and pulped. The puree obtained was enzymatically treated, at 40 Celsius for 1 hour, with pectinase to remove the pectin and centrifuged at 6,000 revolutions per minute for 5 minutes. 5 ml of supernatant was taken to analyze patulin. Patulin was extracted with ethyl acetate and then cleaned up by extraction with sodium carbonate solution. Extracts were dried with anhydrous sodium sulfate. After evaporation of ethyl acetate, patulin was resuspended in water adjusted to pH 4 with glacial acetic acid and determined by reversed phase LC with UV detection. Patulin was detected under UV at 276 nanometers wavelength. Patulin was expressed in ppb equals ngg-1 apple. The recovery rates obtained by spiking apple pieces with 10, 40, and 100 nanograms patulin G-1 in duplicate were 96.2%, 88.5% and 69.1%, respectively. Finally, the limit of detection of the analysis was 5 nanograms G-1. 5 nanograms G-1 or 5 parts per billion is less than 50 parts per billion that meets the requirement of FDA mentioned in the introduction. Although the progress of decay was variable, infection of apples with conidial suspensions resulted in lesions in all the apples. At 1 Celsius, the time for lesions to expand to 2.5 centimeters was approximately 50 days. The time to achieve a lesion of 2.5 centimeters at 20 Celsius was 5 to 7 days. When the data obtained by measuring the increase of lesion size with time in apples were adjusted to Branni's model, Branny and Roberts, 1994, R-squared values obtained varied from 70% to 99%. Figure 1 shows some examples of the nonlinear regression plots obtained at 20 Celsius. Regardless of the other factors, patulin accumulation at 1 Celsius was higher than at 20 Celsius, 217.2 and 199.5 nanograms G-1, respectively, although not significantly. Please view Table 1. Patulin accumulation was affected by inoculum size. However, the INNOVA test suggested that the response to inoculum size was slightly influenced by the temperature. There were some limitations to this experiment. Patterson, 2007, who studied the effect of some fungicides and growth inhibitors in different strains of P. expansum, observed that patulin production was not stimulated in high patulin producer strains and suggested that patulin production may have a limit. During cold storage, P. expansum growth and patulin production does not seem to occur at the same time, being primary metabolism enhanced and detrimental of secondary at initial stages of infection. It seems that production of patulin in cold storage starts when a critical biomass is achieved. Concerning interactions, growth parameters and changes in patulin accumulation observed in me suggest that interactions between different isolates to occur and that these interactions may result in a decrease in patulin accumulation. Either a combat-like interaction or further metabolism of the toxin may be involved in this. Results regarding the inoculum size reflect the importance of hygiene in packing houses facilities but also suggest that interactions between individuals of the same strain may occur. In vitro studies should be carried out to learn more about these interactions. Thus, some phenomena like demarcation of discrete territories or dominance of one of the interactants could be observed. In view of our results, the interactions observed may vary depending on the strains tested or the size of the inoculum used. This experiment showed the way to count the amount of patulin in apple, thus in apple juice. The procedure showed which factors affect its growth rate. Once scientists know exactly how to regulate patulin growth, it will get easier to meet FDA's requirement not to exceed 50 parts per billion of patulin in apple juice.